Well, by my clock, it is 12:30 Eastern, so we'll go ahead and get started. My name is John Mann. I am a postdoc fellow at the North Central Regional Center for Rural Development, and I want to welcome everyone to today's webinar. It's titled New Poultry Vaccine Tech, Leadership for Advanced Responses to Animal Diseases. Before we get started, I want to point out a couple of features. First, to the right of your screen, again, if you haven't done so, we've got a survey asking how you heard about this webinar. If you wouldn't mind taking a moment and responding to that. And about halfway through the presentation, we're going to have a question and answer session, or I'm sorry, we're going to have a trivia question in which we're going to ask you a few questions, and those will be just for fun. And then at the conclusion of the webinar, the conclusion of the presentation, we'll have some poll questions, and so if you wouldn't mind sticking around after the Q&A session for those. Next, we're going to change screens about 3 times during this process. Demonstrate that right now. So if you'll look to the top of your screen, there's a little person with their hand up in the air, and when you click on that arrow a little ways down, there's an applause feature. And so at the conclusion of the presentation, if we could all give Dr. Jackwood a warm round of applause, Adobe Connect style, that'll be much appreciated. And then click screens one more time. At the bottom of the screen, you'll see a chat box. And as we're going through the presentation, as questions come up, if you'll take a moment and enter your question into that chat box, just type right above the word everyone and then enter. And we'll take your questions at the end of the presentation. And with that, I want to thank everybody again for your participation. And now Dr. Jackwood from Ohio State University is going to tell us more about today's topic.
Thank you, John. I want to first of all thank John Mann and the North Central Regional Center for Rural Development at Michigan State University for giving me the opportunity to present this webinar. This is kind of a new thing for me, so I hope it works out well for us, and I guess we'll just see how it goes. The topic today is on a disease that I've worked with for many, many years, infectious bursal disease. And I want to talk to you a little bit about the company that we've started, LARAD Incorporated, which stands for Leadership for Advanced Responses to Animal Diseases, and how we are positioning this company to better produce vaccines and diagnostics for infectious bursal disease virus. So with that, we will get started here. The ideal vaccine, before we can talk about what we're doing to improve the vaccines for bursal disease, let's talk about what is involved with the ideal vaccine, and that is efficacy. Of course, it must work. We would like it to produce a long-lived immunity in the animals. We want it to be stable during storage, economical to produce, and of course, safety is always a factor. So the problem with conventional vaccines is they don't meet all these criteria, and most of them are inadequate in at least several of the different categories. And I will go into that as we proceed with how the vaccines for infectious bursal disease virus are and where they seem to fall short. So infectious bursal disease, this is a very highly acute disease of chickens. The disease is usually seen in 4 to 6 week old birds. It's infectious and can occur in 1 week to 20 week old birds, though. It targets the B lymphocytes. These are the cells in the bursa fabricius that are go on to produce antibodies in the bird. And so because of that, they will cause immune suppression. And if the virus infects birds at a young enough age, it will cause a permanent immune suppression. If the birds are older, and this would be anywhere from 5 to 10 weeks of age, the immune suppression is more transient. It occurs during the disease itself. So this immune suppression is a real problem because, first of all, the birds become more susceptible to other agents, not only infectious agents that normally cause disease, but also opportunistic infections that would normally not cause the birds any problem, but because they're immune suppressed, it can cause quite a bit of a problem in the flock. The other thing that happens is these birds, once they are immune suppressed, do not respond to vaccinations anymore. So they become more susceptible to some of the more important infectious diseases because we can no longer vaccinate for them. Well, there's several different types of the disease, and I want to run through them for you here quickly. This is just a quick picture of one of the subclinical type forms of the disease we have. In this form, the bursas here get very small, as you can see, compared to the controls. This is a bursa fabricius. This is where the B lymphocytes are maturating. And so when the virus infects these, the bursas just get very, very small. The birds, if you look at the flock, they basically do not show any clinical signs of disease. So that's why we call this the subclinical form. And we really don't see any other lesions in the bird itself except for these very, very small bursas. So that's one form of the disease. Another form of the disease is the classic virulent strain. This is the one that we've seen around for many, many years. And this strain, in this particular disease, the birds do show some signs of depression. They are listless, they sit a lot, ruffled feathers, watery diarrhea. So you do see morbidity in this form of the disease. The bursas become very large, they can be yellowish in color, and this happens after about 2 or 3 days, and then after that, the bursas just get small. And other lesions in the birds, we really don't see much of anything with this classic type form of the disease. And then the third type or third form of this disease, the birds become very, very ill. The mortality can be quite high, actually. It can be anywhere from 15 to 30% in a broiler and 30 to 80 or 90% in a layer. The bursas get very large and hemorrhagic. We get lesions in the skeletal muscles. These can be seen in both the breast muscles and the leg muscles. And the disease will sometimes kill the birds so quickly that the bursas don't really show much of a sign of any pathology at all. But mostly with this very virulent form, we see these large bursas with big hemorrhagic lesions in them. So those are the 3 different forms of the disease that we see in the field. Infectious bursas, those are the pathotypes that I just described to you, and then there's also antigenic types of the virus. So the pathotypes are the subclinical pathotype, the classic virulent one that I mentioned, and then, of course, the very virulent one where we have the high morbidity and high mortality. These are all serotype 1 viruses. There is a second serotype 2 of the virus. The serotype 2s we usually find in turkeys and some other birds, but the serotype 2 viruses, up till now, we haven't found any that actually cause any disease. So the disease-causing strains are the serotype 1s. Okay, so the antigenic types of the viruses, there's several different of those. And with the control vaccination programs that we rely on so heavily, these antigenic types are the most important as far as the virus goes. So we have 2 different types, the classic antigenic type, and these are the viruses that have been around since the 1960s. They've usually produced the clinical disease with the large yellow bursas, and this is an antigenic type that everybody is very, very familiar with. The variant types are ones that we initially found here in the United States. They now have spread to many other countries around the world, particularly Central and South America, but we also have found variant antigenic types in other countries, including Europe, South Africa, and Asia. So there's 2 basic antigenic types that most people have, that most people recognize with this disease. But it's really much more complicated than this. When we start looking at the different strains that we find around the world, we find a lot of new antigenic types, and this is because of antigenic drift. Antigenic drift is really the main cause of this, and I'll explain how these viruses change their antigenic structure to these new types in the next series of slides. The surface proteins of the viruses are what's controlling the antigenic type of the virus, and particularly this protein called VP2 or viral protein 2. It's on the surface of the virus and it is in control of the antigenic type of these strains. The other protein that's part of the structure of the virus is the VP3. And this is an internal protein. It's kind of a scaffolding protein, if you will. And the VP2s are built on top of this VP3 protein. There is also a RNA genome. It's a two-segment genome. We're not going to talk too much about that. And there's another protein associated with these virus particles, and that's the VP1, or what is the RNA-directed RNA polymerase. And we're not going to talk much about that one. The most important one for this discussion today is the VP2 protein, because that's the one that's on the surface of the virus. So let's take a quick or closer look at this VP2 protein. This is the physical structure of it. You can see that there are these loop structures at the top of it. There's 1, 2, 3, 4 different loops. And these loops are sticking out on the surface of the virus. And they are what is really the entry point of the virus into the host cell. They're controlling how the virus attaches to the cell. So they're very, very important as far as the virus goes with its ability to infect and cause disease. So naturally, antibodies to these loop structures are going to block that and neutralize the virus. So these loop structures are what's controlling the antigenicity of these bursal disease viruses. Because of the sequencing that we've done, we know what regions of the genome are controlling the different loops. So we can look at the sequence of these viruses, and there's 4 distinct regions. And those 4 distinct regions control the sequences of the amino acids on each one of these loops, and consequently the antigenicity of infectious bursal disease viruses. So here are those 4 regions. We call them regions A and B down here, and then we also have peak 1 and peak 2. These are kind of the antiquated descriptions of these regions. They're actually, the loop structures have specific names now, but we don't need to go into that. Suffice it to say that within a loop structure, at the peak of these loops, there are mutations that are occurring in these viruses. As you can see, the dashed lines here means no change from the sequence across the top here. And when you see a new letter down here, you can see these are new or different amino acids that are cropping up in these different antigenic different viruses that we've listed over here on this end. So each of these loop structures, you can see there's amino acid changes. These are the mutations that are leading to the antigenic drift that we see in the viruses today. The VP2 protein is an interesting one in that it has to appear on the surface of the virus such that these loop structures are sticking out. If these loop structures are not sticking out on the surface of the virus, then we aren't going to have the proper orientation for these antigens. In addition to that, the VP2 comes together as a trimer to form the surface of the virus particle. That is, 3 VP2 proteins come together to form this triangular structure. And when they do that, they actually create some new antigenic sites. These new antigenic sites are created at the junctions of where these VP2s come together. We don't see these antigenic sites if we have a VP2 expressed by itself. But when it's in this form, this trimer form, then we see all the antigenic sites that are on the surface of these viruses. So let's quickly talk about the vaccination programs that are out there now and what's trying to be done. So the breeder vaccination program is the one that's most important, or the one that I wanted to talk about today, because that's where the products for our company, LARAD, are designated for. So the goal of a breeder vaccination program is to provide a uniform, high-quality antibody to the chick by vaccinating the hens. So we're going to vaccinate these breeder birds, and then they're going to pass that antibody that is produced onto their progeny, onto the progeny chicks, through the egg yolk. So we want to get a nice high titer in these hens. And to do that, the best way to do it is to give them a live vaccine when they're young, and then when they're in their laying period, to give them multiple injections of a killed vaccine product. So the maternal immunity, for it to work, it should match the viruses that is found in the field. That's very important. If you're vaccinating the breeder flock with one virus and the challenge virus in the field is a different antigenic strain, your maternal immune program is not going to work. So this doesn't always work because of the mutations that are occurring in field viruses. In other words, if we are vaccinating with a one strain of the virus to our hens, our breeder flock, and the virus in the field ends up changing on us because of antigenic drift, now our vaccination program is not going to work anymore. And we're going to start to see disease in those young birds. So this has been actually corrected or helped by using autogenous breeder vaccines. And those of you in the US will recognize these. These are the vaccines where we go out onto the farm, identify the problem virus, bring it back to the lab, create a vaccine from it, and then send it back to the farm of origin so that they can use it in their breeder vaccination program. These have been quite successful, these autogenous breeder vaccines, because of the antigenic variability that we're seeing in the field, and the fact that the commercial vaccines that are off the shelf usually are antigenically different than the viruses that we find in the field. So these autogenous breeder vaccines have been very valuable to protect against this disease. So what's the problem? So the problem is that most breeder vaccines for bursal disease virus are produced in chickens. You say, well, why is this a problem? Well, it's a problem because the birds are infected in isolation facilities, and the bursal tissues then have to be harvested and prepared for the vaccine. This is quite labor intensive. You can get a pretty high yield of virus, but the process is slow, labor intensive, it's expensive, and sometimes the amount of virus that you get out of these bursas is quite small, because as I showed you in the earlier slides, during the infection with some of these viruses, the bursas just get very small after the infection has occurred. And so sometimes the amount of virus material or bursa material that you can harvest to generate a vaccine might be very low. So the vaccine virus must be inactivated to ensure safety. That's because you're producing these vaccines in birds. It's a live virus and replicating. So once that virus is harvested for the vaccine, it has to be inactivated. And if there's incomplete inactivation, as you can well imagine, that could be a problem. So this is another step in the process of making the vaccine that can be expensive, because you must ensure that it's been completely inactivated. And then one antigenic type of the bursal disease virus is produced with each batch of the virus that's made in these birds. So in other words, we can only protect against one antigenic type with each batch. So if we're going to try and protect against multiple antigenic types of the virus, we're going to have to make these vaccines in several different flocks of birds. So again, this becomes quite expensive to do it this way. So John, I think we're going to take a break at this point. Is that right?
Yeah, we've got a couple of trivia questions. So let me see if I can get these to pop up here. There's the first question. And here is the second question. So if you in the audience would take just a moment and take a crack at these questions. I'll give you about 30 or about a minute. So changing some answers around. about another 15 seconds. So according to my cheat sheet, the answer to question number 1 is actually C. So it looks like about maybe 2/3 of folks got that one. And number 2, everybody got that one right. The answer is A. So let me hide these. And Daryl, I'll let you get back to your presentation.
All right, thank you, John. So the answer to the problems that we're seeing with infectious bursal disease virus vaccines right now, we feel, is virus-like particles. The virus-like particles, which I'll explain what they are here in a moment, but they can be used both in vaccines and as diagnostic reagents. And Ohio State University has filed a provisional patent on this, and our company, LARAD, is licensing that technology, and we'll be making virus-like particle vaccines and diagnostic reagents for infectious bursal disease virus in the very, very near future. So what is a virus-like particle? Well, it's pretty simple, actually. You just take the proteins that are associated with the structure of the virus, and I've showed you those already. This is the VP2 protein that I have here in this panel. You can see this is the side version of it, and this is looking at it from on top. And then we need one other protein that is required for assembly of this of these proteins, and that's the VP3 protein, which I mentioned earlier is the scaffolding type protein. And so when the VP3 and the VP2 proteins come together, they assemble naturally into a virus particle. And the virus particles look identical to the virus that we would harvest if we were harvesting from a chicken or a bursa. The only difference is they lack the genetic material that's in the virus. So because they lack that genetic material, that is the double-stranded RNA genome, they are non-infectious. So this is a naturally attenuated or naturally killed, I should say, vaccine that will not cause disease because it doesn't have the genetic material inside it. But yet the surface of these virus-like particles are identical to that found in the virus, and that's very, very important. The antigenic structure and antigenic quality of these vaccines is extremely important because in years past we have worked on subunit vaccines for bursal disease virus, and the subunit vaccines that have been published and examined by several labs, including mine, usually don't work very well. We get somewhere between 50 and 75% protection using these types of vaccines. And that's because we were only using the VP2 protein. We naturally figured it was the one on the surface, so consequently that's the only one we really needed to use. Well, as I showed you in an earlier slide, these VP2 proteins must form that trimer, that triangular shaped trimer. And not only that, they must be presented so that these loop structures are on the outside of the virus particle, so that these loop structures are available to the immune system. Now, when we do just the subunit part, or just the VP2 by itself, what we end up getting is these tubules. They are round cylindrical tubules, and the surface loop structures of the VP2 end up getting buried inside of those. But when we produce a virus-like particle, what we get is the trimer formed in the right shape and presented in the right way so that those loop structures are pointing out onto the surface of the virus. So here's an example of an electron micrograph of just the VP2 proteins being expressed, and you can see this tubule-like structure that forms, and it just buries the loop structures down inside of it. So this becomes actually a very poor antigen or very poor vaccine. And unfortunately, it appears that my VLP slide over here isn't showing up. These would be the virus-like particles, and they look just like the virus particles that you would see in any electron micrograph. And I apologize for the fact that that picture did not show up on here. But you don't see those long tubule structures that you see with just the VP2. And again, this goes to the quality of the antigen so that those loop structures are sitting on the surface of the virus and available to the immune system. So what is the innovation of this product that we're talking about today? Well, over here on this side, I have what the industry is currently doing. And on the right side, I have what our company is capable of doing using the virus-like particles. Well, first of all, the vaccine in the industry is grown in live chicks. So this is a very, as I said, time-consuming and expensive process. Whereas we can produce virus-like particles in the laboratory. It's very clean, it's very easy, and we can produce them very quickly. When you're making a vaccine in birds, you're really making one at a time. So that we can, if we're wanting to vaccinate against multiple strains of the virus, we would need to make several batches within the birds. Whereas with the virus-like particles, we can make multivalent vaccines very easily and very quickly by including multiple we can include multiple VP2s proteins in the preparation. For the current industry product, because it's a live virus, it must be inactivated, which means additional work to ensure that virus is safe and truly is inactivated. Whereas the virus-like particles, these are safe products from the start. There is no live virus here. There's no genetic material in the virus-like particles, so it is a completely safe product. As I mentioned, producing these vaccines in birds can be expensive and time-consuming. Just harvesting the bursas from all these birds alone can take quite a bit of time, whereas harvesting the virus-like particles from our cell cultures is very economical and very easy to do. And finally, the animal use issue again, and containment issues. When you're growing these viruses in birds, you must use some sort of a containment site or room so that the virus isn't going to get out into the environment. And with the virus-like particles, since this is all being done in the lab, obviously this is not necessary. So our solution, problems that we solve with the virus-like particles. Virus-like particle vaccines can quickly be engineered to protect against the mutating bursal disease viruses. We can quickly change the VP2s that we're using to make a virus-like particle and make virtually any antigenic type of virus that we find out there in the field very quickly and easily. So conventional vaccines do not account for antigenic drift. And the new VLP vaccine antigens can also be used for diagnostic assays. The problem we're having with diagnostic assays is that the diagnostic ELISAs are based on old antigens, very old antigens, mostly classic virus antigens, and there are some that have some variants, but even those variant antigens can be 20 years old. The ELISA tests are detecting antibodies in the progeny usually, or the breeder flock. What we're trying to do with the diagnostic test is identify whether the vaccine worked or not. So we're looking for high-titered antibodies using these ELISA. And if the antigen we're using in those ELISAs is not matching the vaccine that we're using to generate those antibodies, then sometimes our diagnostic assay results can be very misleading. The other thing that virus-like particle technology does is it eliminates this bottleneck that comes with using animals for vaccine production. So we can increase the vaccine supply and meet the end market demand simply by getting away from relying on isolation facilities and growing these viruses up in birds. As I mentioned before, product safety, the virus-like particles are a completely safe product. You don't have to worry about inactivation, whereas the live viruses that are being grown in the conventional vaccines today must be inactivated and then proven that they are inactivated. And finally, lower cost of production for the virus-like particles. This is kind of an obvious one maybe because we're not dealing with live birds, isolation facilities, feed, and all that. We can simply grow these things in the laboratory. A little bit about our business and our business structure. The LARAD Incorporated was it's a very new company. We became into existence in March of this year, of 2013. We are incorporated in Wooster, Ohio. I am the founder and scientific advisor for this company. We have a CEO that we've identified, and that is Mr. Ken Rudd. And he is part of BioBusiness Consultants, his own consulting company, but he has been a marketing manager and marketing director for several vaccine companies in the past and has over 40 years of experience in this area. Because the technology was developed at Ohio State University, they own the intellectual property and they will be an equity partner in our company. That is they will own a small share of the company for the agreement to have a exclusive license to this technology. And then finally, our company is located at the BioOhio Research Park. And the president and CEO of the BioOhio Research Park here in Wooster, Ohio is Dr. Shauna Brummett. And she is our business advisor. She's an entrepreneur with biotechnology business development experience, and she has been extremely helpful in getting this startup company off the ground. So, our company, LARAD Incorporated, the vaccine products that we have for infectious bursal disease now, we have one product that's really what we call off the shelf. This is a virus-like particle vaccine, and it replaces the current breeder flock vaccines that are out there now. So, in other words, we can make a virus-like particle vaccine that is identical to the current breeder flock vaccines that are being marketed by the major vaccine companies. The other product that we have is a made-to-order product. This is the product that is basically like the autogenous vaccines being used in the United States. This one addresses the rapidly mutating virus. So we can go onto a farm, identify the virus that's causing problems there, make a VLP vaccine to that particular strain of virus, and then sell that back or market that back to the company that's having the problem so that they can get it into their breeder vaccination program and get some maternal antibodies in those young birds. And finally, our third product is a diagnostic reagent for ELISA kits. This kind of goes hand in hand with our made-to-order VLP vaccines. We can also make an antigen that matches those field viruses so that when you're using that vaccine in the breeder flock, you'll have a diagnostic kit or an ELISA kit that will exactly match that vaccine, and you can get a better feel for how well your vaccination program is working and what kinds of antibody titers you're getting into those young chicks. Finally, our company has some products in the pipeline, swine PRRS virus, poultry looking at reovirus. We're also looking at some viruses or some diseases in fish and cattle that all could benefit from using the virus-like particle technology. So with that, I will end this webinar. And again, thank you for your attention. And I'm looking forward to hopefully working with many of you. Thank you.
Okay, Daryl, thank you for that. We're going to go ahead and start the question and answer session. So if you've got a specific question, go ahead and take a moment and put that into the chat box. And Daryl, I've got a couple of questions for you. My first is, I've gotten about 15 to 20 emails this like yesterday evening and a couple this morning asking about the international market. So some of these folks are overseas, and so what's the possibility for maybe moving this overseas? I know there's a concern in with a lot of poultry viruses coming maybe out of Asia area. So what would you say about market production for that?
That's one of the areas that we're actually targeting. We feel that that's a very, very important market, very important area for control of this disease. In particular, the very virulent form of infectious bursal disease virus seems to be really worldwide. That's the biggest problem that we're seeing in many of these countries is the very virulent form. We don't have a really good vaccine for the very virulent form of the virus, particularly for breeder flocks. What's being used in the breeder flocks now are classic viruses, and classic viruses are antigenically close to very virulent strains, but they're not an exact antigenic match. Now, as you can imagine, trying to grow a very virulent virus in birds and creating a vaccine using conventional methods could be very problematic. So making a virus-like particle for the very virulent strains of the virus was one of our priorities, and that we've actually done that now, and we have a virus-like particle for the very virulent virus that we're hoping that will be marketed worldwide for breeder flock protection. I think
Okay.
other international problems that we're seeing are the variant viruses, particularly in South America and Central America, and I don't see that is a big issue either. I believe that making the virus-like particles for those strains is simply a matter of identifying the sequences of the viruses that are present in those countries, and we've been doing that for many years simply by submitting samples to my laboratory, identifying the viruses, and we can certainly then create a VLP from those sequences.
Okay, thank you for that. So we've got a question, first question from the audience, and so the question is, what would be the risk of interference between several VLP vaccines?
Okay, that's a good question. With the conventional vaccines, those vaccines are being made by, you make them individually and then you combine them at the end so that you have maybe 2 or 3 different antigenic strains in the killed vaccine. With VLP vaccines, we can do that too. We can make individual VLPs and then combine them into a product at the end. But the nice thing about VLP vaccines and bursal disease viruses, we can actually include the VP2 proteins from 3, 4, 5, 6, or even 7 different viruses in the same VLP. So in other words, those VP2s are all on the same virus particle, and we're not taking them individually and combining them into a vaccine. We're actually putting those VP2s on the same virus particle. The technology allows us to do that, and we've actually done that and shown that they will protect against multiple strains of the virus. So we don't feel that there will be much risk of interference when we make the vaccines that way.
Okay, another question. So the VVP2 is not the only protein responsible for immune response against IBD. Why are other proteins not included in the VLP vaccine?
Okay, so the VP2 protein that's on the surface of the very virulent viruses, and I'm assuming that's what the VVP2 means, the very virulent VP2. That protein is indeed the only one that's really responsible for antigenicity. When we talk about pathogenicity, the ability of these viruses to cause disease, their virulence, that's when other proteins come into play. We know that VP1 is partially responsible for the virulence of the virus. But when it comes to antigenicity and vaccines, the VP2 is the most important one, but that VP2 must be in the correct form and sticking out on the surface of the virus so those loop structures are exposed.
Okay, so the next question, can this presentation be sent to others who have interest in this and join later? And I will address that one. Absolutely. In fact, we are recording this, and so if you're currently watching and you would like to get a copy of this, please send me an email. And like I said, I've received since before we got started, I received about 20 or so emails from folks who would also like a copy to this. So I will be more than happy to pass that along. So the next question, it asks, are you using any kind of adjuvant also? I'm not real clear what that's asking, Daryl. Maybe you've got some more insight.
Yeah, the adjuvant is actually being used. It's the same adjuvant that's being used in the killed vaccines for the products that are on the market now. It's a water and oil type adjuvant, and it tends to work very well with those killed products, and it's no different for the virus-like particle vaccines. So it's essentially the same thing it's there on the market now. We've talked about being able to incorporate virus-like particles into current vaccines that are on the market, and that would certainly be easy enough to do because the makeup of those vaccines is very similar. We just would have to add the virus-like particle into it. So yeah, the adjuvants are there, and it's the same thing that's being used in the market now.
Okay, so the next question is immune response higher against different forms of IBD using this new vaccine compared to conventional vaccines?
The answer to that is actually a no. The immune response is not higher. The immune response is higher quality. In other words, we're matching the antigenic structure of the field viruses with our VLP vaccines. And so the protection is better because the antibody that we're making matches what's out there in the field.
Okay. While we're kind of talking, that question was kind of related to production. I want to throw another one of mine out real quick. And so I'm curious, from your perspective and your experience, what are the potential target firms, companies that are out there to use your product and or be involved with your company? Maybe something relative to the size of the firm or where they are in the actual production chain. In poultry, what do you think about that?
That's a good question. In fact, the virus-like particle vaccines could really replace the conventional vaccines that are being used for breeders right now. So the products being used by Mariel, Merck, Lohmann Animal Health, HIPRA, all those vaccines that are killed products for breeders, we could take their viruses from those companies and make a VLP vaccine for them. It would be identical to what they're using now as far as an antigen goes, and they wouldn't have to make it in birds anymore. Those are some of the companies that we are targeting for these VLP vaccines. Now, when we come to the made-to-order vaccines, those are the ones that are the designer vaccines that are matching the antigenic structure of these viruses that we see here in North and South America, the variants. Those could be marketed by those larger vaccine companies too, but we could also market those directly to poultry producers. Some of the larger farms in particular who use autogenous vaccines all the time could come to our company for those vaccines rather than going to another company to make the virus in birds. We could make the autogenous, or we could make the virus-like particle vaccine for them as a made-to-order designer vaccine. So we're hoping to work with all these companies when it comes to the designer vaccines. And then individually, we'd like to also work with the large vaccine companies, as I mentioned, to possibly replace the products that they have currently on the market with virus-like particle vaccines.
Okay, thank you for that. So the next question from the audience, what expression system is used to produce the VLPs?
We are using the baculovirus system. There's a number of different systems that could be used, but we found that the baculovirus system folds the VP2 protein correctly, which is very, very important. And it produces it in extremely large amounts so that the production costs are lowered. So the baculovirus system is the one we're using.
Okay, next question from the audience. Can we, and I'm assuming this is from like at a production side, so can we revaccinate birds? Oops, I lost the question. Let me get back to it.
Can we revaccinate birds during production using this new vaccine is the question. And yes, it can be used exactly like what's being used now in the field. These killed vaccines for breeders are given multiple times during the laying period of these breeder flocks to keep their immunity high and keep those antibodies being transferred through the yolk to the progeny in a high enough amount so that those birds are protected for the first 2 or 3 weeks of life. So yes, you can use these VLP vaccines to revaccinate breeder flocks during the laying period.
So a follow-up question with that, can this vaccine be mixed with other oil emulsion vaccine like the triple ND, IBV, EDS vaccine?
Yes, absolutely. You could include these virus-like particles for bursal disease and those mixed products so that you would have a mixed bursal disease virus, Newcastle disease, infectious bronchitis virus vaccine that you could use. So yes, absolutely, they can be mixed in with those other agents.
Okay, so I have a question kind of from an economist perspective, and this is the first thing I thought about given your presentation. But so regarding a company, who would be maybe an appropriate person to kind of participate with what you're trying to accomplish? Kind of beyond the maybe teaming up with some of the larger organizations. What other kinds of things are you actually looking for in the context of your actual company and trying to go forward? Like what would be the next step?
Our next step is actually to start working with some of these larger vaccine companies so that we can replace their products. So we're looking for contracts with those companies so that we can actually begin to start producing these virus-like particles and have them either send them through their testing procedures or look at our data and get these out onto the market as soon as possible. We're looking to work with investment firms, angel investors, or any investment firm that's looking to capitalize in a startup company. So we need capital, obviously, for space and equipment and things like this, and we're already starting to generate some of that through these investment groups. But that's always obviously on our radar, is to find someone who's willing to invest in a new startup company. And our advisor, Sean Abraman, who is the president and CEO of the research park here on campus, is really taking the lead role in helping us with that. Did that answer your question, John?
I think so. That they got to the kind of the next step in what you're seeking. So it looks like we got a couple more really good questions. So the next one is, can VLP production, not the VLP itself, be regarded as genetically modified by the EU? That's an interesting question.
Yeah, that's a good question, and I guess I don't have a good answer for that because I'm not sure what the EU qualifies or identifies as a genetically modified agent. The agent that we're using to express these proteins is baculovirus. So that baculovirus is a recombinant organism. It produces the protein, but once we have the virus-like particles produced, the baculovirus is inactivated, and so that recombinant organism is no longer part of the product, if you will. It's simply a protein that is being made, and so the end product, as you said, not the VLP itself, and that's true. The VLP itself is just a protein that was made through the recombinant baculovirus. Whether or not the recombinant baculovirus would be considered regarded as genetically modified by the EU and come under those rules and regulations, I don't know. It's part of the process of production, but it's not the final product that is being used in the birds.
Okay, the next question, another good question. It says, can this technology be used for avian influenza vaccine production?
Yes, it can. Virus-like particles have been made for influenza already, for other species, and it certainly could be done for avian influenza too.
Okay, here's another question I appreciate as an economist. Is this vaccine actually cheaper than conventional vaccines?
It's cheaper to produce, yes, because we can make it in the laboratory and we don't have to do it in isolation rooms by infecting birds. So the whole harvesting process, inactivation, and quality assurance that goes along with inactivation, we don't have those problems. So it is cheaper to produce this vaccine, yes.
Okay, so follow-up comment about the question with the EU. There's a thank you, and then it says knowing the boy, I'm not even going to try to pronounce that. I guess I will try. Baculovirus.
Baculovirus. That's what I've been saying, John.
Thank you. Yeah, is recombinant, gives me some clue about the regulatory risk. So, well, I kind of hit on all of my questions. I see that there might be one other person typing in. Daryl. Oh, here we go. So let's talk a little bit more about the economics. So you said it's cheaper to produce. Maybe think about, if you could expand a little bit on the cost side relative to what you're doing versus what you might see when you actually sell the product. So maybe talk a little bit more about the economics and relate that to maybe the technique, your production techniques.
So the production techniques are really twofold. First of all, you have to produce the recombinant baculoviruses, and that takes maybe 3 to 4 weeks to do that. Since the VP3 protein is the same for all virus-like particles, you don't need to reproduce that one. The only one you need to do is the VP2 protein, which is going to be on the surface. So if you've got an antigenic change in the VP2, that's the protein that then needs to be cloned and incorporated into baculovirus so that you can produce it. Once that's done, then it's simply a matter of growing these baculoviruses in the cells and harvesting the proteins that they produce. And that's a relatively easy thing to do. These viruses, baculoviruses, grow in insect cells. They actually grow; those cells grow at room temperature. They can be grown in suspension, so you can do these in large vats and produce liters and liters, tens of thousands of liters if you wanted to, relatively easily. It doesn't take quite that much because baculovirus is an extremely good expression system, and so we can get quite a lot of protein per mil out of these cultures. So a 50-gallon fermenter would grow more than enough vaccine virus and certainly make it very, very inexpensive compared to growing that same amount in birds. So it is a very economical technique. Now, once the protein is produced, harvesting it is relatively simple. You simply harvest the cell culture fluid. That is where the virus-like particles have assembled. And then it's just a matter of inactivating the baculovirus, which can be done by a number of different methods, but it's, again, a relatively simple procedure, and including the vaccine then into the adjuvant. And once it's mixed with the adjuvant, it's ready to go. There's no concentration needed or anything like that, and it's quite economical process to do that. So from that point, then it's simply bottling and shipping, which all vaccines have.
Okay, thank you. Thank you for that explanation. Go ahead, I'm sorry.
I'd like to mention about the economy of this, John, is that when you're making these virus-like particles, the product that you end up with is very, very clean compared to a product that is coming out of the chicken. These bursa-derived antigens, as they call them, where you're grinding up bursas and mixing them with a diluent and then the adjuvant, they can contain quite a few different things, not necessarily other infectious agents, but you may actually have some bacteria there sometimes, which again gets inactivated when the virus does. But it's a much dirtier product than what we find with the virus-like particles, because the bursas have so much more lipids and proteins and all in them from the tissue that we don't have when we grow these things. We grow the virus-like particles in cell culture.
Okay, here's a couple of technical questions about the products. So in what degree should the vaccine be kept?
And I think the next one, what's the shelf life, is also related. And the shelf life of these is very good, actually. We were concerned because when we previously worked with just the VP2 protein subunits alone, they were not very stable. But when you get these things into a virus-like particle, that structure becomes extremely stable. It's resistant to proteases, and we find that at room temperature, that virus-like particles were actually stable for an 8-week period at room temperature. We saw no difference in the potency and the structure of the virus-like particles after holding them for 8 weeks at room temperature. We did the same experiment at 37 degrees and held them for 8 weeks at 37 degrees and got the same results. The virus-like particles were even stable for 8 weeks at 37 degrees. Now, we haven't tested them beyond this 8-week period, but what we feel is if we can get them out at least 2 months worth, that would certainly be enough for shelf life as vaccines are being turned over and sent out from the production line.
Okay. Thank you for your responses to all these questions, and thank you in the audience for your questions. I think that might be it. What I'm going to do is I'm going to click over to the next screen, and if you've got a couple of questions, if you still got a question, the chat box will still be open, and we'll stick around for just a couple of minutes. So before we completely wrap this up, if everybody would just take a moment and find that applause feature, and let's give Dr. Jackwood a warm round of applause, the Adobe Connect style. And again, the next webinar we've got coming up in the series is on May 22nd with Dr. Lance Skiddeman at University of Illinois, and we're going to be talking about biofuel. So if you've got an interest in that, there are a couple of links towards the bottom of the screen for the NCRCRD page, also to register, and I've got my email and also Dr. Jackwood's email included, and that's also on the previous screen. So again, if you would like a copy of this, please send me an email and I'll be happy to forward you the link. And with that, I don't see any more questions popping up, so Daryl, thank you so much for participating. I really appreciate that, and those of you who participated in the webinar in the audience, I really appreciate your participation. So thank you very much.
Again, John, thank you for putting this together, and thanks to the North Central Regional Center for Rural Development. I really appreciate the opportunity to do this.
We were very glad for you to participate. Thank you. So for you in the audience, I'm going to leave this page up for quite a while, so you can go ahead and take your time to respond to these questions. And with that, I think we'll call a close to the end of this webinar.